Okay, so we're back again. So we had our Benedict solution that tested for simple sugars. No, that's not how you spell it. Benedict solution. Okay, so that was our simple sugar. And so you want to remember that one. And I can't spell right now. I need to have another coffee apparently. Okay, so that was important. Then we want to go on now to test for complex sugars. And we just do one test that we use over and over again throughout the semester. And hopefully you remember this experiment where we tied off a diffusion bag and dropped it into water. Do you guys remember we did that for the diffusion lab? And if you remember what we put in the bag, we put in glucose and starch. So that was a really, I mean, it's kind of a clever experiment because then outside you can see what was able to travel in. So we actually put iodine outside and it was able to cross that barrier of the bag and go inside, and that meant that all the starch that was at the bottom turned black. Black or purple, like kind of a really cold colored purple. So those are the complex sugars. So the way that the complex sugar starch links together is just in long chains. And so we want to know that that's a disaccharide, but they're even longer. So you just keep going, linking them together, and there's always an oxygen in the corner. You're gonna have carbons and oxygens, and then some H's dangling off. But all of these link together, and you can actually link them in like large nets of molecules. Obviously, I'm not a very good chemistry drawer. So, but I want you to realize how monster huge this molecule is. Okay, so we'll just connect some more. Here we go. Well, you can't really do that to an O. Sorry, that has to be a carbon. We're gonna link more up here just to make it even bigger. Okay, so off the screen, huge, right? So this is our polysaccharide. So poly means many. So polysaccharide. And there's two C's. Okay, so polysaccharide is going to be starch, and that's our complex sugar. It's this large network of monosaccharides. So each one of these is one, which means mono. And so you glue all those together with a hydrogen bond. It's actually called dehydration synthesis is what glues them together and just leaves the oxygen in there. But you glue them together and have this big, big molecule. And the point of the diffusion lab was that this big, big molecule was too big to leave the bag. So that was really important. The iodine could go in and dye the starch purple, and it couldn't leave the bag. The starch was too big to leave. So we use iodine to color starch black. And sometimes it does come out kind of purple depending on what we've put in the mix. But you want to know that starch should be black or like a dark purple color, and I'll try on the exams to always refer to it as black. And iodine is kind of like a light amber color. I don't know any other examples other than amber. It's like a light brown. When you spread it on the skin, it's kind of yellowy, orangey looking. And then you put it in the starch and it's a distinct color change. So if it's just iodine and water, it should be kind of an orangey light brown. And when it's a positive result for starch, which cornstarch is a definite positive, right? So, I mean, you're saying starch in the name of cornstarch, so you should be pretty right on, knowing that that's your positive control, and see that coal black color form. And then when you compare that to the polysaccharide starch from potato. So potato, we know they're starchy vegetables. So when you have the potato in there, you should have known that that's your starch. Right there, potato. So hopefully that makes sense. That's our polysaccharide. We knew intuitively that potato should have starch in it. We also tested cornstarch just to make sure that we knew the positive result. And that should be good. To test a polysaccharide, you use iodine. So thanks.